good morning. This is Krsna Rajesh. I am a third year BSc Biotechnology student from Amrita School of Biotechnology. I am currently doing an internship under Dr.Dayana in Pushpagiri Research Centre. So today we visited the animal house. So here the animal house is acclimated to CCSCA, which is the Committee for Control and Supervision of Experiments on Animals. So CCSCA has given license for only experimenting on animals and not breeding them. Therefore, the animals are obtained from a licensed breeder. These animals are then quarantined for around 7 days in a quarantine room. So the quarantine room helps in clearing up contamination and disinfecting the animals. So once the animals are quarantined, they are then transferred to the animal room. The animal room may be a rat room, a mice room or a rabbit room, depending on the animal being used. So next step is taking these animals to the surgical room, where the experiment takes place. So the surgical room has two parts, a preparatory part and a surgical area. So the preparatory area is where the animals are being prepared before the experiment, and the surgical area is where the actual experiment takes place. So before the experiment, the animal is given anesthesia. So there are two options of anesthesia here, gas anesthesia and injected anesthesia. So gas anesthesia we use isoflurane, and for injected anesthesia we use ketamine xylazine. So there is an isoflurane apparatus being used here. So after the experiment, the animal is then transferred to the post-surgical room. Here is where the animals are being kept for the entire duration of the experiment. So once the entire experiment is completed, the animal has two options. One, it can go for rehabilitation or it could be euthanized. So euthanasia is a method being used here. So carbon dioxide euthanasia method is being used using a carbon dioxide chamber. So these animal carcass and the waste is then disposed of using a yellow bag and then incinerated. So the animals, this is what happens in an animal house. So the animal house has a one-way transport. Basically there is a fixed entry point and a fixed exit point. There is a food dip in both the points, which is filled using an antibacterial solution, which is a primary disinfectant method. So the basic instruments there is in an animal house are exhaust, there is a dehumidifier, an AC, blood analyzer, serum analyzer, there is an autoclave, weight balance, water purifier, water mist extinguisher. There is also something called an automatic light dark system, which gives 12 hours of fixed light and fixed darkness. It is a 6 AM to 6 PM procedure which is being fixed here in the animal house. So these are the basic instruments in the animal house. So the basic studies being conducted here are biocompatibility test. Then there is toxicity study. We have wound healing, bone regeneration. The future prospects are analgesic and anti-inflammatory activities of novel compounds. So coming to how do you actually propose an animal study? So OECD guidelines are followed to submit a study proposal to the IAEC, which is the Institution of Animal Ethics Committee. So once the study proposal is being approved by the IAEC, only then can you conduct a particular animal study. So in vitro analysis report of the compound is submitted using the OECD guidelines to IAEC. So here there are two types of study, acute toxicity study, which is followed by the 423 OECD guideline. Here we use 6 animals. There are 2 steps, 3 animals each. And then there is a sub-acute toxicity test, which is OECD 407 guideline. It is a 20-day oral toxicity, 10 rats per group, 5 males and 5 females. There are 4 study groups here, control, low dose, mid dose and high dose. 10 rats each, there is a total of 40 rats. Then an example of a study conducted is analgesic study. There is a read and heat method, which is also known as clean flip method, and we also have a hot plate method. See here, there are 6 rats per group and there are 4 groups, control, standard, low dose test compound and high dose test compound. So there are a total of 24 rats here. So the number of rats are fixed according to the guidelines. So this is what happens during an animal study proposal, and what happens in an animal house has already been discussed. So thank you.